Evening, everyone. So after some weeks, we are back with one more workshop on drug designing. It's a part of drug designing. As it we call it QSAR modeling. That is quantitative structure activity relationship. Okay, so we actually predict the biological activity of any kind of compound molecule, or you can say a test molecule, to find their biological activity as a broad, and then we can analyze further into molecular docking as well as molecular dynamic simulation. Okay, so and this is a part of drug designing, and it's very integral to find out the biological activity of any kind of compound, because if you have a ligand molecule, or if you have a compound molecule and you do not know the exact bioactivity, then you couldn't perform molecular docking into your particular receptor. Okay, so in that case we just need to find out their biological activity, and this quantitative structure activity relationship modeling is very important. Actually, in the previous workshop as well, we were dealing about, you can say, admit analysis and a lot of other things. You all are familiar with molecular descriptor as well, if you have attended those workshops. So in case of QSR modeling, we just go through different compounds and their biological descriptor, what we call it molecular descriptor. You can say in the admit analysis as well, we have screen different compounds, or you can say drug molecules, to find their bioactivity through different other tools as well. For example, PASS online and all that thing. Okay, in case of PASS, it's an online tool, and it predicts the biological activity of any kind of compound molecule based on the structural information. Okay, spectral information, particularly NMR spectra. Okay, but in case of QSR modeling, we just need to get the, you can say, experimental value of any kind of you can say enzyme inhibition. For example, if you are working on amylase enzyme, and if you want to find any kind of drug molecules, then you just need to find the IC50 value, or you can say LD50 value of any kind of compound molecules that targets that particular enzyme. What we call it here, it is if you take amylase enzyme, then you need to find the IC50 value of those compounds into amylase. Okay, in that way we can just calculate it. Okay, we can just prepare the model and we can test it in our compound. Okay, so let me present first of all, and I am Suman Dungal, Managing Director of Nepomics Biotech, and today I will be guiding you through the entire workshop, and if possible we will also conduct one demo session by your presence as well. Okay, so let me present about QSR first, and then we will go on to the demonstration session. Okay. Let me share the screen. Okay. Okay, let's move on to the introduction of QSR. Okay, so QSR stands for Quantitative Structure Activity Relationship, and we actually predict the biological activity of any kind of compound based on their chemical structure. Okay, based on their particular structure, we predict the biological activity, and it's a basic, you can say, modeling tool. And we use different kind of molecular descriptor. I've already told you. So mathematically, it is expressed as the activity of any kind of molecule is the function of those physiochemical properties, or you can say molecular descriptor. Okay, in this case, you can see on the right hand side as well, there are different kind of molecular descriptor mentioned: log p value, molecular weight, hydrogen bond donor, hydrogen bond acceptor, topological surface area, and many more. Okay, we can use this kind of you can say biological descriptors to calculate the biological activity, or biological activity of any kind of test molecule, test compounds. Okay, so in case of QSR modeling, so where does it actually fall in CAD workflow? Okay, so let's deal with that first. So as we all know that in case of drug designing, we first need to identify the target. Target means, you can say, we actually do molecular docking or any kind of you can say drug designing thing on protein, actually a protein or any kind of enzyme, or you can even perform on DNA. So DNA, or you can say nucleic acid or protein, can be a target. Okay, so first of all you need to identify the target, you need to validate its structure, or you can say if the target structure is known in the, or it is present in the database, then you can go through structure optimization, or if it is not present, then you can move on to homology modeling. Okay, and then you can optimize the structure, and then you can find the target binding site, or you can say pocket. In the particular receptor, you will find the active site residues over there, and into that active site residues, or into that active site, you need to perform the molecular docking. You need to target that particular spot. Okay, in case of enzyme molecule, you can find multiple binding site as well, so you need to find the actual you can say feasible region where your ligand is going to bind. Okay, so after finding that pocket, you just need to optimize those pocket structure as well. For example, molecular dynamic simulation can be performed to exactly find the dynamics of those pocket as well. Okay, if it is not possible, then there are other methods. Okay, so you can do that, and then after verification of those targets, you can just dock your compound molecule. Okay, so ligand can be obtained through different mediums. Okay, we have already mentioned about it. So you can design your ligand by yourself, or you can go through different databases to exactly you can say accumulate those ligand molecules, and then you can optimize the structure, energy minimization, and everything. Okay, and then you can prepare the library of your compound, and then you can perform molecular docking into the target. Okay, and then you can just optimize those complex. After performing molecular docking, you will get a lot of different conformation. Okay, and then based on the binding energy and as well as interaction pattern, you can just rank those conformation, and based on the two different factors, as I've already told, two different factors, you can just rank them, and you can allocate the potential heat based on that result, and then you can do molecular dynamic simulation. Okay, and then you can perform biological assay, and then you can go on to QSR. But actually it is mentioned after biological assay, but you can perform QSR modeling before molecular docking. Okay, so 2D QSR, actually two-dimensional QSR is done before molecular docking. Okay, we can perform 3D QSR after molecular docking, or you need to perform molecular docking and then only you can do 3D QSR. But in case of 2D, you can just perform 2D QSR, know the biological targets, and then you can go to molecular docking. Okay, that is the prime, or you can say that is the major difference between 2D and 3D QSR. We'll talk that later as well. Okay, so once the biological targets are known, you can do molecular docking and go on to perform other stuffs. Okay, like molecular dynamic simulation and all that thing. And if you couldn't find the potential heat, then based on the information of QSR as well, you can go through pharmacophore modeling. You can change the derivative, or you can produce a different derivatives over there, and then you can just continue the process. Okay, so in this way we actually design the drug. And QSR, you can do QSR modeling just before the molecular docking. Okay, to find the exact target, you do not need to lose time before molecular docking as well. So you just need to do 2D QSR before that, finding the biological target of your ligand, and then go to perform molecular docking. Okay, in this way we can exactly fit QSR into the workflow. Okay, CADD workflow. So the importance of QSR. Okay, let's talk about the importance of QSR. So if you know the biological activity of compound before performing molecular docking, then you can save time. Okay, you can save ample amount of time, and you can also save plenty of you can say financial burden as well. Okay, you can save your money. Okay, actually. So if QSR is done before molecular docking, or if QSR is done before going on to next step of drug designing, then you can easily find out the target. Okay, actual targets. Okay, based on the experimental data, and with that you can easily go to perform molecular docking or any other stuff. Okay, that is what is important. That is very important for these two things. Okay, and next is optimization. So you can optimize your compound using QSR. Okay, for example, you'll know the biological activity of compound, so you can screen out different other compound molecule that is not feasible into the target. Okay, so in that way you can find the potent compound. Okay, and you can go to perform you can say biological assay with a lot of confidence. Okay, and next is you can predict the biological property early. Okay, so actually the main purpose of QSR is to find the biological property before molecular docking. Okay, so if you can do these things before molecular docking, then you just get the information about you can say admit parameters as well. Okay, or you can get the information about the fit. Okay, how fit is the molecule. Okay, so if you find that early, then it will save you in the later stage of drug designing. Okay, so there are three different components in QSR. The number first component is biological activity. So we need to actually get the actual biological activity of test compound, or not test compound, you can say the compounds that you are going to prepare model for. Okay, so in QSR we actually you need to prepare a model first. Okay, so to prepare a model you need a lot of compound molecules, so you need to get the exact experimental data of those compounds. Okay, as I've already told you, IC50 value. Okay, IC50 value of those compound molecules in that particular target. For example, if I'm doing molecular okay, if I'm targeting aspirin, okay, so I need to find the IC50 value of aspirin in whatever I'm doing. Okay, for example, aspirin is functionally active on you can say cyclooxygenase COX-2 or COX-1 receptor. Okay, so I need to find the IC50 value of aspirin against cyclooxygenase. Okay, COX-1 or COX-2. So in this way you need to just find the biological activity of aspirin and a lot of other derivatives. Okay, so we will talk about it later. And next is molecular descriptor. So the molecular descriptor, there are a lot of molecular descriptor actually. Previously before in 1990s, or you can say 1980s, people used to use Hans equation for QSAR modeling. Okay, we'll talk about the chronology later. So people used to use this kind of equation where their lesser amount of molecular descriptor were used. Okay, and it was not actually explored as well. So later in after 2000, people changed this you can say Hans equation into modern you can say regression analysis tools, and now they analyze different kind of descriptor, number of descriptor. Okay, even more than 20 or more than 50 even. Okay, based on the number of compounds that you are selecting for the model preparation. Okay, so in case of molecular descriptor, like you can go for molecular weight, you can go for log p value. Okay, whatever biological biophysiochemical properties you can get it. Okay, you can just get that information from databases as well, or even you can read a paper and get that information, and you can just allocate those or you can just accumulate those molecular descriptor and then prepare a model. Okay, that is what is required for QSAR modeling. And next is mathematical model. So once you get a biological activity of those compound molecules, okay, and the molecular descriptor of those compound molecules, you need to prepare a mathematical model. Actually you need to just do the regression analysis of molecular descriptor and biological activity. Actually molecular descriptor are kept on x-axis, and biological activity is kept on y-axis, and we need to perform a regression analysis. Okay, in case of 2D QSR we just can do linear regression. Okay, and then if you are using ML or any other tools, okay, 3D QSR, you just you can go through plenty of molecular descriptor, and you can easily calculate you can easily prepare the model. And these deep learning tools can also help you do the thing. Okay, for example, in case of machine learning, you can use artificial neural networks and all that thing to get the exact model. Okay, so we'll talk about 3D and this ML thing later as well. Okay, and then once you calculate once you do the regression analysis, you will get a significance of your statistical, or you can say statistical significance of your result as well, and then you can use a formula to calculate the IC50 value of those models, and then you can also predict, or you can predict your own IC50 value as well. Okay, so it's a Hans equation. Okay, so it was used back in 1980s. Okay, now we don't use Hans equation because you can see log p value over here. Okay, they just focus on lipophilicity of a compound. Okay, so they focus on lipophilicity. That's why it is not much valid as well. Later on I will show how lipophilicity of any kind of compound impacts the IC50 value of it as well. Okay, it is not too much reliable. Just by taking lipophilicity, you cannot just evaluate the IC50, or you can say evaluate the biological activity of any kind of compound molecule. So later on they changed this as well. And you can see log I 1 by Siemens, you can just say it as IC50 value, okay. And you can see here log P and sigma and ES value and so all that thing. So sigma stands for electronic constant. They use electronic constant, or you can say they use electronic stability and all that thing as a molecular descriptor. And they also they used to use a static hindrance of those compound molecules as another molecular descriptor, okay. And based on these molecular descriptor, they used to calculate the IC50, or they used to calculate the biological activity of any kind of compound molecules. Later on it has been modified already, okay. So let's look at the modern equations now, okay. Modern QSAR model, it is a QSAR model based on linear regression, what we call it MLR QSAR model as well, okay. Or you can say descriptor based QSAR model, because we use number of descriptor in this modern QSAR model, okay. You can see in the equation as well. Y means, you can say, biological activity or IC50 value of that particular you can say ligand against any kind of receptor, okay. And A stands for intercept, okay. Intercept, the value of intercept, you can get it from regression analysis. After doing regression analysis, it will give you one output file. In that you can easily get the intercept value, okay, Y intercept. And next is x1, x2, x3, okay. You can see x1, x2, x3 and so on. These are the molecular descriptor. For example, if I take you can say if I take molecular weight as one descriptor, you can name it as a x1, okay. Log P as a x2 and so on, okay. It depends upon your own you can say analysis, your own requirement as well, okay. What kind of molecular descriptor are you going to use? It depends upon yours research work, okay. And next is this Bn, okay. Bn value, they stand it stands for regression coefficient, okay. For example, the regression coefficient of each and every molecular descriptor, like B1 is for the coefficient of x1, that you can say find that you can get in through regression equation, okay. And then later on you can just calculate the IC50 value, okay, predicted IC50 value actually. So this is the thing. It is a modern QSAR model, okay. And let's talk about the evolution of QSAR, okay. So back in 1960s, they used to use Hans equation, okay. And then later on it was modified to parabolic Hans equation. And it is particularly okay, that hasn't been changed, okay. They just focus on log P only, okay. Before that it was electronic constant and static thing particularly, but later on they just focus on log P, which was not reliable, and it is already been discarded as well, okay. Nowadays people use modern linear regression QSAR model, because it is more reliable than the traditional one. And it has also been evolved to different other you can say models of QSAR, like 3D QSAR, okay. In case of 3D QSAR, you rely on a molecular docking results, okay. And then you need to have a molecular alignment as well. The conformation of molecule needs to be exactly similar, okay. So in case of 3D QSAR, but in case of 2D, you just need to allocate, or you just need to accumulate those different compound molecules. You can do that, okay. And in case of machine learning, if you have a number of plenty of molecular descriptor, if you want to evaluate you can say plenty of molecular descriptor into a single run, then you can use ML thing, okay. So let's talk about the workflow of QSAR, okay. First of all, you need to identify, okay. First of all, if you have a test compound and you don't know the biological activity, then you just need to go through the structure of that compound, okay. What is the structure of that compound? What is the scaffold, or what is the basic unit of that compound, okay. So you need to identify the scaffold. Once you identify the scaffold, so scaffold means, for example, if you are doing work on phenol, then benzene can be the scaffold of it, okay. So you need to find the scaffold, and then based on that scaffold, you can go to you can say you can go to the database and then collect number of those scaffolds, okay, and their derivatives, okay, and then you can get the data from those databases. Like in our case, we use Campbell, okay. So you can just type the you can say for example, if I'm doing some research work on compound A, and I think like it is active against acetylcholinesterase, then I just go to Campbell and search acetylcholinesterase with the same kind of scaffold compounds, okay, and then I will get a lot of compounds that has been active against acetylcholinesterase, and based on that I will collect the data, okay. So that is the thing. So first of all, you just need to find what kind of compound you are doing, okay. So based on that, you need to identify the scaffold, and then you can go to source your, you can say, predicted biological activity. For example, if it is active in that particular biological target or not, okay. So in this way, you can just go to Campbell and type the name of your target, probable target, and then you will get the scaffold of your compound, okay, test compound, and based on that you can get the IC50 value of different compounds, okay, and you can put a filter over there even, okay. There can be a compound web that has been approved by FDA, or that is in a clinical trial as well. So you can just select those compounds, okay. It doesn't mean that you just need to find FDA approved compound only, okay. So you can get a compound or you can get IC50 values of any kind of compound molecule that is in a clinical trial, okay. So because even in case of clinical trials, okay, people have performed a lot of experiment, and the wet lab result is there, and it is already a good compound, okay. So you can just check that, okay. And then once you pick the molecules, you just need to find the IC50 value of that particular compound into the receptor, okay, into that particular target. And the most important thing here is that you just need to find the IC50 value of that compound with the same kind of biological assay, okay. For example, if you are doing anti-cancer activity of any kind of compound molecules, and if it is done by one method, then other compounds needs to have the same kind of assays as well, okay. So if the assays is different, then you cannot select them, okay. So keep it in mind about that as well. And next is, once you gather the information, or once you collect the data as multiple compounds with IC50 value, then you just need to find out the molecular descriptor, okay. So in case of those compounds, you will get a lot of molecular descriptors, okay. You just need to select some of the key molecular descriptor. Later on I will tell you how to select it, okay. It depends upon what you are doing, and it also depends upon how you are analyzing the data, okay. So the best way to select the molecular descriptor is to select molecular descriptor based on ADMET parameter. For example, molecular weight, it signifies the size of compound, okay, size of compound. So the larger molecules is hard to penetrate the cell membrane, okay. So in this way you can select molecular weight as you can say permeability test, or in case of log p value, it is significant for lipophilicity, and it is directly linked with fat solubility, okay. So it signifies the membrane permeability, absorption, distribution, all that thing, okay. So select based on different ADMET theme, okay. For example, one descriptor can be selected from absorption specific or absorption related function, another can be selected for distribution, and so on, okay. So in this way you can select the molecular descriptor, and later on you can build the model for it, okay. Taking molecular descriptor in x-axis and experimental, you can IC50 value. Actually it's not IC50, it's PIC50. Later on I will clarify that, okay. The IC50 value on y-axis and molecular descriptor on x-axis. You just need to do the regression analysis, or you just need to do the regression thing, and you will get the data, okay. And then based on that data you will get a coefficient, x-intercept, coefficient of x variables, and all that thing, and then you will just calculate the IC50 predicted IC50 value by your own model, okay. So once you prepare a model, you just need to validate that as well, okay. So for example, the IC50 value of any kind of compound molecule in experiment is found to be 8.1, then your based on your model it should be around 8, okay. you cannot get an exact, sometimes you will get it as well, but the value needs to be around 8. Okay, so in this way you can validate the structure or validate the model as well. Okay, and you can test that model into your experimental compound later. Okay, so this is it about the presentation. Okay, so if you have any kind of queries, before moving on to demonstration, I want to solve it. I want to hear it about the queries, and then we will move on to another, okay, so demonstration thing. Okay, so if you have any question, then you can arise, or we will move on to demonstration. Okay, if there is no any question, then let's move on to demonstration. Okay, so I will take aspirin, okay, aspirin and its derivative, and I will prepare a model based on the, you can say, biological activity of aspirin, that is COX receptor, as I've already told you. So it has an analgesic property, and it fits into the cyclooxygenase receptor as well. So let's do that. Okay, let me share another screen. So just a minute. Okay, and let me share. Okay, so here is the, okay, let's see the demo first. Okay, so in here I have selected 16 different compounds. Okay, aspirin and indomethacin are FDA approved drug, and these all are the compounds I have obtained from Campbell database. Okay, and they do not have their name because they are in clinical trial. Okay, so in different stages of clinical trial, 1, 2, or 3. Okay, so I have obtained those compounds. So actually the my test compound has a scaffold of aspirin. Okay, so if I'm testing any kind of compound molecule that is related to aspirin, then I need to prepare a model based on the scaffold. Okay, so aspirin is a scaffold here. Okay, so aspirin and their derivatives, I can easily you can say collect those compounds and then prepare a model. Okay, so that's the basic idea. So in case of aspirin, the PIC50 value. So PIC50 value is a different thing than IC50 value. Okay, we can calculate PIC50 value using this formula. Okay, so later on I will let you know about it in the Campbell as well. So in the Campbell you can see the standard, you can say standard value of any kind of compound molecule, and based on that standard value you just need to calculate the PIC50. Okay, so later in another column you can see the P Campbell IC50 value, that is supposed to be PIC50. Okay, you can either copy that PIC50 value, or you can even convert through the standard given over there. Okay, so you should not write the units here. I've just used this unit here, but you shouldn't write it because PIC50 doesn't have any kind of units. Okay, so I've just copied the bioactivity of these compounds as well. So the bioactivity needs to be similar. So these all have the bioactivity, similar bioactivity, same bioactivity, actually not similar, same bioactivity. And next is target. So they all target on the same receptor, that is cyclooxygenase. Okay, so the PIC50 value has been obtained over here. So let's move on to the molecular descriptor now. So I need to allocate some of the molecular descriptor of these compounds. Okay, so aspirin, indomethacin, and so on. So here you can see 16 different compounds and a small string as well, and the molecular descriptor of those compounds has been given over there. For example, I have taken molecular weight, log p value, and topological surface area. I've told you about selection of you can say molecular descriptor. So I've selected on 3 different categories. Okay, for example, log p value, it signifies the lipophilicity of these compounds that is directly linked with fat solubility, and that determines the membrane permeability, and it also plays role in absorption, distribution, and even not metabolism, but particularly absorption and distribution. Okay, and in case of molecular weight, so molecular weight directly indicates the size of any kind of compound molecule. So it actually acts as a you can say indicator of permeability. Okay, permeability, or you can say penetration. Yeah, that's a permeability. Okay, so the larger molecules are less permeable and the smaller molecules are easily permeable to the cell membrane. And next is topological surface area. So topological surface area, it signifies the area covered by polar atoms of that particular compound, and it particularly indicates absorption, blood-brain barrier indicator as well. It is an indicator of blood-brain barrier as well. And next is, it also signifies the hydrogen bonding activity of that particular compound. Okay, hydrogen bonding activity of particular compound. So these has different roles. Okay, so based on these roles, I have selected this parameter. Okay, you can select number of different molecular descriptor, but it has certain you can say guideline as well. For example, the number of compounds. Okay, the number of compounds that you are going to prepare a model is greater than or equal to 5 into molecular descriptor, number of molecular descriptor. Okay, for example, here I have used 16, or you can say 15, 16 compounds. Okay, 16 compounds, and there are 3 different molecular descriptors. So 16 is the compound number of compounds. 16 is greater than or equal to 5 into 3, right? 5 into 3, that means 15. Okay, obviously 16 is greater than or equal to 15, so it satisfies that particular you can say guideline as well. Okay, so based on that, you can select the number of compounds. Okay, how many compounds do I need to select would be done through the number of molecular descriptor. For example, I've already told you about it. Let me make it clear as well. 5, or you can say number of compounds is greater than or equal to 5 into number of molecular descriptor. Okay, so that's where you can select you can allocate the number of compounds as well as a number of descriptor in your particular model. Okay, and next is I've copied the PIC50 value from the Campbell. Okay, that was already shown as well, and I've used the source as well, and later I plot that, or you can say I calculate the regression analysis, or I've done the regression analysis of these compounds using PIC50 value and molecular descriptor, and I've got the result. So in x-axis you can just plot these molecular descriptor, and y-axis, or you can say as a y component, you can use PIC50. Okay, so you can go to data. Okay, if you do not have data analysis, in some cases, if you have not used data analysis before, then you will not get that. So you can just go to file, and you can go to options, and you can go to add-ins. Okay, and then you can go to Excel add-in, go. Okay, you can get a here you can get the analysis toolpath. You just need to select it. Okay, you just need to press OK here, and you will get the data analysis toolpath there. Okay, so in this way you can do that. And next is to get the information of regression, you just need to go to data analysis. Okay, and then you can go to regression. Okay, you can go to regression, and then you can just in y-axis you can just select this. Okay, till here, and then in y or in x-axis, or in x-axis you can just select molecular weight to topological surface these data. Okay, in y-axis you need to select PIC50, and in x-axis you need to select this molecular descriptor. Okay, once you do that, you will get that result. You just need to allocate the output range as well. Wherever you want to put this summary output file, okay, you just need to allocate. For example, if I have allocate here, so I've got the result. You can do it in a similar way. Okay, if you press OK here, then you will get the similar kind of similar kind of output file. Okay, so here you can see. Okay, so let's talk about this data right now. So here the regression statistics or the output file has been generated, and the R square value is quite good. Okay, 0.83, and the adjusted R square. So we need to look for the adjusted R square actually, not R square. Okay, so adjusted R square means it has already, you can say, it has already analyzed the data based on number of descriptor and number of, you can say, lipophilicity, or number of, sorry, PIC50 value. Okay, so if you have enough data over there, you will get a nearby you can say R square value. Okay, it is an adjusted R actually. So the value needs to be similar. Okay, for example, here I can see 0.83. Okay, and next is 0.79. It is close enough. So if you have not selected the, you can say, proper molecular descriptor, number of molecular descriptor, as I've already told you, the number of compounds should be greater than or equal to 5 into number of a number of molecular descriptors. So if you have not satisfied that thing, then you will not get a R square, adjusted R square in this kind of format, or in this kind of ratios. Okay, so the ratio needs to be exactly feasible to the equation. Okay, so that is the thing. And next is the significance. Okay, significance needs to be 0.005, less than 0.005. It is quite lower, so the statistical it is significant because we have taken enough data. Okay, so that is the thing. And next is intercept. You can get the value of intercept over here. So I have copied the intercept value here, and then the coefficient of variable x variable 1. So in case of my data, x variable 1 is molecular weight, x variable 2 is log p value, and x variable 3 is topological surface area. So I have allocated them based on the information given here. So x1, x2, and x3. Okay, so we need to use this information to calculate our you can say calculate our predicted IC50 value. Okay, so let's go to calculation now. So here it is. For example, in my data I have used 16 different compound, and the actual IC50 values has been given here, and the predicted one. So predicted one, we need to use this equation to see how the data has been or you can say distributed. Okay, so the IC50 value of PIC50 value of aspirin is 4.82. Okay, so once I prepare a model, I found out that the IC50 value of aspirin is 5.02. Okay, so how did I calculate it? So let's talk about it. So the formula is here. Okay, PIC50 value. To predict PIC50 value of any kind of compound, we use this formula: A plus B1 x1 plus B2 x2, and so on. Okay, so there are three different you can say molecular descriptors. So x1, x2, and x3 are satisfied over here. A stand for intercept, so 3.1, and B1 stand for the coefficient of x1, you can say molecular descriptor, and B2 stands for coefficient of x2, and B3 stands for coefficient of x3. Okay, so in this way we calculated it, and the aspirin has the predicted value of 5.02 and so on. Okay, you can see the values lying over here. Okay, and we just need to plot this plot the scatter diagram as well, scatter plot as well. So to plot that, you just need to place actual PIC50 value on x-axis and predicted value on y-axis. Okay, and you will get this kind of information. Okay, so if the regression is fine, okay, it's R square is 0.83, and you can see okay, you can see the ideal model. Okay, in case of ideal model, the x or you can say x value needs to be equal to y value. Okay, that is the ideal one, but actually we do not get ideal one, but the value, or you can say the the core needs to be diagonal in shape. Okay, so it's quite you can say matching as well. So I can easily say that my model is better. Okay, so in this way we can allocate, we can predict or you can validate our model as well. Okay, next is you need to plot predicted as well as residual IC50 value as well, PIC50. So to calculate residual IC50 value, you just need to okay, you just need to calculate the difference between actual and predicted one. Okay, so actual minus predicted, you can get the value over here. Okay, it is not nanomolar, sorry, we cannot use the unit in PIC50. So in this way you can plot it. Okay, and the ideal curve says that the value lies in the values should be in this zone. Okay, 0 zone, or you can say the value lies in this linear axis of 0. Okay, so it is quite fine as well. Okay, ranging from minus 0.6 to 0.6. So next is, okay, you can also correlate your data. Okay, so correlate your IC50 value with molecular descriptor actually. So I have done one correlation analysis. Okay, I've checked whether the log p value directly impacts the actual IC50 value or not. Okay, so in this case, okay, I plot log p value on x-axis and actual IC50 value on y-axis, PIC50 value on y-axis. Okay, so after doing that, I got this information. Okay, so actually this information signifies that log p alone cannot say that, okay, the value of log p cannot determine whether the compound is you can say active in that particular receptor or not. Okay, because you can see the regression is very low. Okay, so we cannot say that log p has that important role for change in IC50 value of any kind of this particular compounds. Okay, so you can even plot for you can say what we have done. Okay, molecular weight. Okay, next is topological surface area and so on. Okay, any kind of molecular descriptor that you have used. Okay, you can just plot that. Okay, and then see whether it is correlated with your predicted IC50 value or not. Okay, in this way you can just link these kind of thing. Okay, and if you have a confusion like, okay, so IC50 log p has no any impact on increase or decreasing value of PIC50, then you just need to think that, okay, log p is a sole parameter. It doesn't it didn't signifies the role of this compound in cyclooxygenase. But if you consider the number of different molecular descriptor, then you can see it has been impacted. Okay, and we can also get the data from here as well. In actual IC50 it's 4.82 and predicted it's 5.02. That means it's quite similar. Okay, so in this way you can validate the data as well. And now talking about how you can use this model to analyze your compound. Okay, for example, in my analysis, I have taken this compound. Okay, it is a aspirin derivative. Okay, so I want to know whether this derivative is active against cyclooxygenase or not. Okay, so to find out the thing, I use its molecular descriptor, 3 different molecular descriptor. We need to use exactly the same molecular descriptor that we have prepared in our model. Okay, so I have used molecular weight, log p, and topological surface area. Okay, so these are the data of this particular derivative. Okay, I can get this from databases like PubChem or Camel even. Okay, so I have used this descriptor here, and I calculated the predicted IC50 value, PIC50 value of, or you can say predicted IC50 value using the same regression curve. Okay, the A value is similar, B1, B2, and B3 are similar. That has been obtained from, you can say, the model itself. Okay, so after calculating it, I found out the PIC50 value was 7.94. Okay, the PIC50 value was 7.94. So to see whether my compound is active against cyclooxygenase, I need to go through the same same data, okay, to check the value and same data. So it was 4.9, 7.9, sorry, 7.9. So you can see the actual IC50 value first, okay, 4.8, 8, 6.1. So my compound falls in between, falls in the range of indomethacin, okay. That means it has a great binding activity against cyclooxygenase, okay, because the PIC50, the greater the value of PIC50, the more binding nature, or you can say more biologically active the compound is, okay. So in this way you can check whether it is feasible or not. So in case of IC50 value, the lower the value, higher is the activity, okay, in case of IC50. But in case of PIC50, the higher the value, higher is the biological activity, because it is calculated in the negative logarithm form. I have already told you. So if you have log p value, or sorry, if you have IC50 value in molar, then you can easily use this equation to calculate PIC50, okay. So this is how we can predict the biological activity of any kind of derivative, okay. So let me open this Campbell, so it will help you out. Okay, it's a Kimball database interface. So I have used this already. So you can get the results over here. So I have searched cyclooxygenase in this search engine, or you can say space. Okay, and then I got this information. So in the left hand side, you can use different filters. Okay, you can use the filters, and then you can easily get the information. Like on the left, on the right hand side, you can see a number of different compounds, okay, and then the IC50 value has been given over here. You can just go through it, okay. Like uh, let me say select, okay, let me see. Okay, you can see the IC50 value over here, okay. Where is the standard? Okay, standard value is present over here. IC50 value 89. So it's IC50 value, okay, it's not PIC50 value. But here you can see P Campbell value. This is PIC50, okay. 7.05 is a PIC50 value. So in this way you can just copy this, okay. And about the AC, you will get the information of AC here, okay. AC description, you can see inhibition of human kidney, whatever thing, okay. Selectivity, okay. You can easily get this two IC50 value from this particular interface, okay. You cannot just copy the IC50 value of this one and this one, okay, because the assay are different, okay. So in this way you can select it. For example, assay format, here it is assay format, it is a protein format, okay. You can go onto the left hand side, and you can you can check it, okay. Assay format, you can just put a filter over here. There are three compounds for assay format, okay. Assay format, you can, but the thing is these two are similar, but the essay description here has been given different than the rest two one, okay. So you can select this along with this two, okay. So that is the another clue for selecting how you can select compound and IC50 value by using Campbell, okay. So in this way you can do it. Okay, let's prepare one model, okay. You can use for practice, you can just use 3 or 4 compounds, okay. Forget about the significance of data. You can just copy 3 or 4 molecular descriptor, or three or 4 compound, and then you can check it, okay. So let's do it. So I'll give you some time. You can just go to Campbell and search the biological target of any kind of compound molecule that you are interested in, and you can just copy it, okay. So let's do it. Just go to Campbell, okay. I'll share the link as well for Campbell. Okay, Rajendra, we cannot share the slide. Okay, you can get the information on YouTube. Okay, you can later check our YouTube channel and then see those information. Okay. So I have shared the link of Campbell. So you can go to Campbell and you can just search on Campbell search bar. Okay, like any kind of receptor or any kind of biological activity that you are interested on. Previously I searched for cyclooxygenase, so you can also do for cyclooxygenase as well. And once you search cyclooxygenase there, you will get plenty of compounds over there. Okay, and then select one of that compound and go to one pie chart given over there. You will see IC50 value there, and just press on that IC50 and you will get plenty of compound. Okay, and you can easily select the IC50 value or PIC50 value from there, and based on PIC50 value you can also select the you can say molecular descriptor there, and you can just prepare a model. So here is given for different compound. The IC50 value has been given over here in nanomole. Okay, and then there is a standard units as well, so you can convert from here, or you can just copy this P Campbell value, PIC50 value, you can say it. You can just copy here, for example 7.05 in this case. Let me put a filter. Oh, let me share it again. Okay, so here you can see I have searched for cyclooxygenase. Okay, and I've got this information, and you can see on left hand side the lot of information are given. For example, if you are targeting mammal, you can just select it. Only a mammal is given here, you don't need to select. And next is assay format. So let's go to assay format. Okay, there are three compounds for it, and here you can see there are 5 compounds, approved compounds. In phase 2 you can see 9 compounds, and in preclinical you can say 178 compounds are present over there. So we need a multiple compound, so you just can select preclinical here. Okay, so first of all let's see about the approved one. Okay, so in case of approved one there is only one. So let's select preclinical. Okay, although it has been shown 5. Okay, okay, because of this. Okay, okay, now you can see. Okay, you can see this compound. Okay, the IC50 value has been given over here. The standard value is 100. You need to convert it if you want to convert by yourself to get a PIC50 value, or else you can just copy from here even, like this is a P Campbell IC50 value, means this is a PI IC50 value. Okay, PIC50 value, so you can just copy it 7 here, but make sure it has been done on the same assay. Okay, so inhibitory activity against prostaglandin GH synthase. Okay, it is the same one. It is protein, it is a assay, so you need to select the assay. Okay, these two are similar. Okay, you can get the information here, but the IC50 has not been given over there, so you can select this, you can select this, okay, 5.51, 5.89, you can select both of them. Similarly, okay, you can select these as well, okay, you can select this, you can select these as well. You cannot select this, you can select, okay, you cannot select this, you cannot select this, okay, you can select this, okay, based on what you are doing, okay, you just need to select the particular assay format, okay, then description of assay as well, what kind of condition they have done their research on, okay, for example, this has been done on human platelets prostaglandin, so the previous data was also done on the same thing, okay, so if you are targeting any other thing, then you can just, for example, if you copy the information of this particular SEEP seminal field, vesicle, sorry, so you just need to go through the same type of description of this tissue based format, okay, so in this way you can copy the IC50 value. So once you copy the IC50 value, so you can also get the information of molecular descriptor here. Let me do that thing as well. So you just need to click on that particular compound, okay, and you can just scroll down, you can get a canonical SMILE formats, okay, and later on at the bottom you can see, you can get the information like molecular weight, topological surface area, log p value, rotatable bond, whatever you want, okay, whatever kind of molecular descriptor you want, you can get the information from here. So I've copied molecular weight, log p value, topological surface area from here, and then created a model, okay. Similarly you can do the same, okay. Once you copy the information, you can go to Excel sheet and then plot the value, okay, IC50, PIC50 along with molecular descriptor on the x-axis and IC50 value on y-axis, and you can just get the regression curve, or you can say regression output file, okay, and based on that regression you just need to calculate the predicted IC50 value, okay. Let me share the Excel sheet again if you are confused with it. Okay, let me share. This is the Excel sheet. So once you get the IC50 value, you just need to copy here, okay, and along with the compounds, name of those compounds. I've used aspirin, indomethacin to FDA approved drug, and rest of all of the things are, all of the compounds are in clinical trials, okay. So I've just copied it and along with PIC50, and don't use this unit, okay, because PIC50 it doesn't have any units, so you just need to do the thing, and then and the molecular descriptor, okay, you just need to copy the molecular descriptor from the same database Campbell, molecular weight, log p value, topological surface area, and so on, and you can just place your PIC50 value here, and then you can plot it, okay, you can plot it here and then get this result, and then x variable, y variable, x variable 1, x variable 2, x variable 3 can be obtained from here, and next is intercept as well, and use this information to and then calculate the predicted IC50 value from this equation, okay. It is a modern linear regression curve, you can say formula, okay, to calculate that particular modern curve QSAR model. So in this way you can do it, and then plot the actual and predicted value, and plot predicted and residual value, and if for residual you can just calculate the difference between actual and predicted, and then get the residual one, and then the residual and predicted and residual predicted and residual value needs to be around 0, so it's quite good as well. And next is you can just plot the molecular descriptor, saying each and every molecular descriptor with actual PIC50 value, and then see whether your molecular descriptor is linked with PIC50 value or not, or you can say if it is you can say impacting the value or not, okay. In this way you can do it, and later on for any kind of compound molecules you are doing, or any kind of derivative you are targeting for, you can just copy the molecular descriptor first, and then the name or whatever thing, and then you can calculate the PIC50 value of that particular derivative, okay, using the same equation and using the same information from the regression, the previous regression, okay, that you have used to prepare your model, and then you can just get the value. Okay, you can convert it into nanomolar as well using this formula. PIC50 value equals to minus log 10 IC50. Okay, log 10, it should be in JavaScript. So in this way you can do it. Okay, if you have prepared, if you have copied your details of molecular descriptor, then you can just share your screen and show me. If any of you are preparing it, if you have prepared your model, then you can share the information here. Let's see how your model is behaving. You can take any kind of test compound, okay, any kind of derivative. If you are confused with anything, then you can just let me know, okay? Okay, could you explain what's the significance of plotting this graph and how to interpret those graphs? Okay, I think I have already told you about how we can interpret the data. So in case of predicted and so let me share it again, okay? That would be easier. So here you can see it is the main curve, okay, actual and predicted value. So you can see how actual and predicted value lies, okay. For example, even you can see from the data as well, the actual IC50 value of aspirin is 4.82 and the predicted one is 5.02, okay. So there is not such kind of rule that okay, you need to your value needs to be similar, or your value needs to be in the range of something or something like that, okay. There is no such strict rule, but make sure your value is quite similar to the actual PIC50 value, okay. And next thing is that you can see in the curve as well. So ideal curve, I've already told you, in ideal curve the x value needs to be similar to y value, or x equals to y is the ideal curve, okay. But if your value is in the diagonal line, then that means your model is excellent, okay. That is how you can calculate it, how you can interpret the graph. And in case of residual, predicted to residual as well, your value needs to be around 0, okay. If it is quite scattered all over the data, then it's not good, okay. And next, it is just experimental thing, okay, whether a single you can say molecular descriptor is impacting your biological activity or not, you can get the information, okay. And based on this information, based on the information of molecular descriptor, you can go through pharmacophore modeling and change your overall, you can say substitute or function group over there, okay. So that is the thing. And apart from that, we can also do 3D QSAR, okay, and we can also use Python programming language to calculate to calculate the regression curve, okay, or to do the regression analysis by using Python. You can screen plenty of molecular descriptor over there, okay. So I think I made it clear, okay. Okay, thank you, Anand. So if anybody has prepared a model, please share it, or we will end the session. If you are doing, then please share it, or we can end the session. You can follow the video that has been already live on YouTube, okay. So you can follow the video and create the model, and if you get any kind of problem, then you can send an email as well, okay. We will deal on that. We will help you out how to analyze or how to obtain the good model as well, okay. So that is a that is all about QSR modeling. So next we'll organize similar kind of program in the future as well. Many sorts bioinformatics program, okay, especially focus on drug designing. So if you are working on a pharmaceutical science, biotech, or even any kind of life science courses, or even in chemistry or pharmaceutical chemistry, then you can follow us, okay? You can follow our YouTube channel. You are already linked with the email as well. So we will keep on updating about the upcoming workshop. You can just attend our program and you can get benefit in your research work as well, and you can share your ideas in here as well, okay? We can also work in a collaborative basis. So if it is possible, then Ananda, can you tell me what you are working, or where you are working? Maybe you are from India, so can you share your research experience, or what are you doing there?
Hello, sir. My name is Anand Bakril. Currently, I'm a faculty at Golden Gate International College. So, even though my works are currently not related to molecular docking or QSR modeling, I am interested in learning about this for my future research.
Okay, okay. So I've been seeing you for quite long time now. So I've seen your presence in the previous workshop as well. So thank you for supporting us. Thank you for attending it, and we'll give our best to help you out. Okay. So let's talk with Supratha. Okay. Are you am I getting the correct name, Supratha?
Ah, yes, sir.
Okay, can
Very
you
good
just?
evening, sir.
Oh, okay. Good evening. So can you just tell me what you're doing and where you're from?
I'm from India, sir. I'm currently pursuing masters in pharmaceutical chemistry from National Forensic Science University,
Okay.
Gujarat,
Okay.
India.
Mhm.
Uh, I'm currently a student. I'm just learning about molecular docking QSR model. Just I'm trying to put these works in my thesis work. That's
Okay.
why I'm learning those skills.
Okay. Are you doing thesis there
Yeah.
currently?
Thank you, sir. Yeah, actually, I'm currently in a first year, sir. Now I have to start, so I'm just looking to molecular docking and QSR models, everything.
Okay, okay. So you can follow us on YouTube. Okay, there we have uploaded a lot of information, a lot of, you can say, tutorials, workshop as well. So in the near future as well, you can join in this kind of program. Okay, and if you're interested in, you can say, uh, any kind of workshop for at least 1 or 2 months, then you are also welcome for that. Okay.
Okay, thank you, sir. Thank you.
Okay. Okay. So let's wrap this workshop for today. So we will be in touch in the future through, you can say, YouTube or any other platform. So you can easily find us there, and hope to see you in the future program as well. Okay. Thank you. Thank you, everyone. There, and hope to see you in the future program as well. Okay. Thank you. Thank you, everyone.